Hello students, this is session number 39 under Molecular Biology chapter for a B.Sc course, Chemistry Paper 8, Biochemistry. This video is under DCE, Digital Learning System, and I am Dr. Veena from Maharani Science College for Women. In this particular session, I am going to tell you about genetic code, definitions, general features, and definition of transcription and translation. So you are going to learn here what is genetic code, what exactly is its definition, and general characteristic features of genetic code. And as I told, the content is about the definition of transcription and translation. We will take one by one now. See, all of you know very well, DNA is the genetic material. Okay. So, the genetic information on the DNA is transcribed into RNA. Okay. And then translated to precise amino acid sequence of polypeptides or proteins. Now, the relationship between the nucleotide sequence of the mRNA and the amino acid sequence of the polypeptide is called as a genetic code. So, please understand this. So, what is genetic code, students? The relationship between the nucleotide sequence of the mRNA, that is messenger RNA. And you know, on a polypeptide chain, you will have amino acid sequence. So, the relationship between these two is known as genetic code. You call that as a genetic code. I hope you have understood what is exactly the genetic code now. See, early genetic experiments conclusively proved that every amino acid is coded by a set of 3 nucleotides on the mRNA. Okay, and it is true also. Now, see, first this genetic code was elucidated by 3 scientists, Marshall Namberg, Robert Holley, and H.G. Corona, for which they were awarded a Nobel Prize in 1968. So, the set of 3 nucleotides on the RNA molecule that encodes a specific amino acid in the protein, you call it as a codon. So, what is codon, my dear students? The set of 3 nucleotides on the RNA molecule that encodes a specific amino acid in the protein is called as the codon. So, what are the general features of this genetic code now? So, this genetic code is a triplet language. Okay. So, genetic code is a triplet code. What do you call it as a codon? Okay. See, for 4 bases, you know 4 bases you have learnt in your previous sessions. So, 4 to the power of 3, 64 codons that encode 20 amino acids. Okay. So, this simple mathematics you should know. Now, remember, genetic code is specific, means every codon codes for a specific amino acid. So, you call it as a genetic code is a specific in nature. And the codons are degenerate, means several codons code for a specific amino acid. Okay. And remember, genetic code is universal in nature. It is universal in nature. And the codons are non-overlapping. The codons are non-overlapping. So, this they will ask you for 4 marks. It's a 4 marks question for you in this particular chapter. What is genetic code and what are the general features of genetic code? So, how do you write your answer? You know the definition of genetic code by this time. So, genetic code is specific and the codons are degenerate and the genetic code is universal in nature and the codons are non-overlapping. Okay. Now, The codons are continuous. Okay, they are not interrupted. So they are continuous. And remember, the first codon is always AUG. So it starts with the first codon AUG. So this AUG is called as the initiator codon. ಏನಂತೀರಾ ಇದಕ್ಕೆ initiator codon. See, it encodes the amino acid methionine. So every amino acid, it is very specific. So hence the first codon on every mRNA is AUG and the first amino acid in every protein is methionine. So here you should understand the first codon or the initiator codon is always AUG, which codes for what? Methionine, which codes for methionine. Now, see, out of 64 codons, only 61 codons encode amino acids. How many left out? 3. Isn't it? See, these 3 codons do not code for any amino acid. Remember, they don't code for any amino acid. So, what do you call them as? You call them as nonsense codons or terminator codons or stop codons. So, where are they found? They are found at the 3' end of mRNA. So, which are those nonsense codons or terminator codons? We use our terminator codons or stop codons. UAA, UAG, UGA. I repeat, UAA, UAG, UGA are known as terminator codons. They'll ask you for 2 marks. What is initiator codon and terminator codons? Okay. See, in every session I'm telling you, I'm stressing out which are the topics and for how many marks you get particular topic. Okay. Now, see, this is a start codon is always AUG, codes for methionine. So you have UAA, which is a stop codon, and this is a polypeptide chain, N terminus chain and C terminus chain. So you find the stop codons at the 3' end of the mRNA. So what you have learned? Definition of genetic code, general features of genetic code. Now, coming to simple definitions of transcription and translation. What is transcription? The process of synthesis of RNA molecule. The process of synthesis of what? RNA molecule complementary to a gene on DNA molecule is known as transcription. Okay, that is, the synthesis of RNA molecule from DNA is known as transcription. And which is the enzyme responsible for this process? RNA polymerase. RNA polymerase is required for this process to take place, and it takes place in the nucleus of every cell prior to protein synthesis. We'll learn another definition, translation. Now, from DNA, RNA is synthesized, which is transcription. From that RNA, that is, the process of synthesis of a protein molecule from RNA. So, you have RNA which is synthesized from DNA. If a protein molecule is synthesized from RNA, that is known as translation. We call this as translation. So, where does this take place? It's the cytoplasm of every cell during protein synthesis. So, here you should know 2 definitions. Definitions for transcription and definition for translation. So, see here you have DNA. From DNA, the genetic information present in DNA. Now, from that, there is a synthesis of RNA. This process is known as transcription. Right? The genetic information contained in a segment of DNA is transcribed into RNA molecule by a process known as transcription. See, during transcription, what happens? An enzyme system converts the genetic information of a segment of DNA into an RNA strand with the base sequence complementary to one of the DNA strand. So I hope is that clear. The process of transfer of genetic information contained in what segment of DNA is transcribed into RNA molecules by a process known as transcription. Okay. So I told you in the previous slide, translation. From this, the synthesis of a protein is known as translation. So this figure will clearly tell you the definition of transcription as well as translation. Now, which are the types of RNA involved? You know very well. There are three types of RNA. One is mRNA, that is messenger RNA, transfer RNA, which is written as tRNA, ribosomal RNA written as rRNA. So, which are the three types of RNAs? mRNA, tRNA, and then rRNA. See, transfer RNA is an adapter molecule that reads the information encoded in mRNA and transfers the appropriate amino acid to the growing polypeptide chain during protein synthesis. So you should know the job of mRNA. So it carries instruction from DNA to the rest of the ribosomes. And it will tell you what kind of protein they should make. So that will give a message that is the job of mRNA. So you see here, this is the job of mRNA, right? So start codon is AUG, right? And it gives a messenger what type of protein has to be synthesized. That is, see, these are different codons which coding for particular amino acid. So this is done by mRNA for the synthesis of the required protein, right? Now, see, I told you in the previous slides, AUG codes for methionine, similarly for glycine, similarly for serine, similarly for isoleucine, glycine, alanine, and when it comes here, UAA, it ends, so it is a stop codon. Okay. So what did you get from all this? You got a primary structure of a protein which are joined by peptide bond. So all in all, what you should know here is tRNA is an adapter molecule, I told you, that reads the information encoded in mRNA and transfers the appropriate amino acid to the growing polypeptide chain during protein synthesis. Okay. Now, what does this rRNA do? This ribosomal RNA molecule associate with proteins to form ribosomes, which is a site of protein synthesis. Okay. So, which helps in the protein production. So, this tRNA gets the right parts to make the right protein according to the instruction of what? mRNA. See, it has got this claw-like structure and you have amino acid attachment site here. Here an anticodon is there here, okay, in this loop. See, here amino attachment site, methionine, which is the first amino acid, okay, that is the, which codes for first its amino acid, methionine. Now, this is the structure of ribosomes. You have two sides, because this I will explain in the next session where you study about translation, the process of translation, that is the synthesis of protein. So you have a large subunit, small subunit, P site and then E site. So what you have learned here is the genetic information contained in a segment of DNA is transcribed into RNA molecule with the help of different types of RNAs. Okay? And this process is known as transcription. So as I already told, the three types of RNAs are involved here. That is mRNA, tRNA and then rRNA. Now, a small doubt you may get here. Then what is the difference between replication and transcription? Isn't it? Because in the previous sessions I've told you what is replication. See, replication and transcription differ in one important aspect. During replication, the entire chromosome is copied to yield daughter DNAs identical to the parent DNA, isn't it? Whereas in transcription, DNA can be, that is, a transcription of DNA can be regulated so that only genetic information needed by the cell at a particular moment is transcribed. Okay? So, specific sequence on the DNA called regulatory sequence indicate the beginning and end of the DNA segments to be transcribed and also which DNA strand is to be used as a template. So, this is the major difference between replication and transcription. So, in this session, what you should know is the definition of genetic code, the characteristics of genetic code, number 1. Number 2, definition of transcription. Number 3, definition of translation. So, from this session, I hope you have understood all the definitions and the particular function of different types of RNAs. Hope you have understood this from this session. Thank you. See? Okay. So, learn the definitions and what all I have told in this session. Thank you.